And the next speaker of our workshop will be Viktor Petухov from University of Copenhagen, Denmark. Viktor will present the results regarding case control analysis of single cell RNA-seq studies.
Hi, yeah, I tried to share my screen. Does it work?
It works.
Nice. So thank you so much for inviting me. And yeah, I will be presenting case control analysis of single cell RNA-seq studies. It's what we do in the lab in the University of Copenhagen in collaboration with Harvard Medical School. So let me start with describing the problem. And the point is that during the recent years, single-cell RNA-seq become higher and higher throughput, and it is cheaper and cheaper. So now we can do lots of cells, and they don't have to be from the same sample. We can profile multiple samples at the same time. And that allows us to do case control analysis, case control experiments. So our data looks like a bunch of samples from control, some samples from case, like in this example, it's our study where like 8 versus 7, or sorry, 10 versus 9, but it can be different samples. There are studies like with 50 samples each side, there are studies with 3 samples each side. And then we want to take some joint space with all these samples. We want to look which samples come from which condition, and we want to do some analysis on that. And there are many questions which we can ask, like did some cell type change their expression in a similar way, or whether some genes coordinated their changes, and basically anything about expression patterns which depend on our case or control study, like a condition, can be studied, and we want to get as many information as possible. But more specifically, given the single-cell RNA-seq is kind of implicit tool to investigate everything, we want to show it explicitly. We want to do more biological experiments and show like real biology. So, but that is like expensive, that is timely. So we want to narrow down the focus, and we want to understand which subtypes should we focus on and which genes within specific subtypes are the most affected. And that would give us like real understanding of the system. And what people currently do is they take some samples, so they put them in a joint space, and you can see that if you just put it like in one space, you will see severe batch effect, because of course there is a difference between case and control. But different tools were developed to remove this batch effect, like Sura, like Konos from our lab, like SVI. And so people align samples in like to remove batch effect. Then they do joint annotation to understand what cell types are actually there. And then for each of cell types, they do differential expression to get a list of differentially expressed genes, and to do some functional annotation on these genes, and yeah, get gene ontology terms. Some papers also do cell type proportions, where they take each subtype, see fractions of cells from a subtype from one condition and from another, and you can see it on the picture. Though honestly, for this particular study, don't completely trust them, because it mostly look like they just did the wrong annotation, because it too much depends on the case or control. And if you look at the papers, most of them follow exactly this workflow. So here are 5 papers like from top journal like Nature Science, and all of them do exactly this. And my claim in this presentation is that it is actually not enough, because we have 2 main questions, which are what cell types are the most affected and how exactly are they affected. And this kind of analysis barely answers any of them. And because if you want to look like what cell types are the most affected, people often look at the total number of differentially expressed genes per subtype. But that is actually wrong, because the number of D genes severely depends on the number of cells, as you can see on the picture. It also depends on the inner variability within the cluster. And basically, total number of D genes, if you don't control for these parameters, only shows you power of your test, not affection of a cell type. So that's not a proper measure. And, if you want to understand how they are affected, looking at differentially expressed genes also gives you very few information, because there are hundreds of them. And you like hundreds of them per cell type. And if you look at total number, it can be thousands. And just looking by eyes on the list of genes, it doesn't make much sense. So people often do some cherry picking, like picking 5 genes out of 3,000 and claim that they know these genes and that these genes explain everything. But of course, it's just a cherry picking. And so, I mean, it's fine that you can find some genes, but it doesn't reveal you the whole picture. And even if you reduce number of genes with gene ontology terms, then you still have hundreds of geoterms. And looking at all of them, I mean, it's much easier than using like thousands of genes, but still not too much sense. And another problem which I need to mention here is that all this analysis severely depends on the annotation of cell types. So if you annotate only like major subtypes, then your power of the test will be very low, and you will like capture only the major changes. And if you go deeper and deeper, if you do like very granular annotation, then your problem will be that some subtypes will be not precisely annotated, and you will have some like various errors, and especially on the border, it can really affect your analysis because some genes are expressed not where they're supposed to, but it's not a problem of genes, it's a problem of annotation. And I personally spent a couple of weeks when we did such analysis just trying to clean the annotation, but it's around impossible because you have like 20 samples, 40 subtypes each, and making it perfect is really hard. And when you go to hundreds of samples, then it's actually impossible. So annotation is also a bottleneck. And what we propose here is a bunch of solutions, which we divide into several separate types. First is gene expression analysis, where we analyze how genes changed, like how cells changed their expression patterns. Second is compositional analysis, where we see how proportions were changed, because it's often the case that maybe some cell type changed its abundance, but not gene expression, or the opposite. You see exactly the same abundance between condition, but expression patterns are different. So these are two major categories, and we also split them to cluster-based and cluster-free. Cluster-free is where we developed the methods to actually do exactly like the same things, but without any annotation. So first we find interesting regions, and then we try to understand what these regions actually are. And I will go through this one by one. Let me start with composition analysis cluster-based. And the very basic thing which we can do is to look at proportion of specific cell type per sample. And then we can plot these proportions for each subtype, and then we would have distribution of proportions because we have multiple samples, and we compare these distributions between conditions. And we can estimate significance, and we can analyze that, and that's fine. But the problem is that it's not exactly enough because of this situation. Look at the two examples at the left. And on the top, you can see that the red and the blue subtypes, so here like numbers of samples, and you have 3 subtypes per sample, and you have 2 groups. And you can see that red and the blue subtypes did not change their abundance, but the yellow did. And on the second, it's the opposite. Red and blue changed a lot, but the yellow is exactly the same. But when we normalize on the library size, when we estimate fractions, these two pictures would look the same or very similar. And moreover, difference would be kind of smoothed because of the normalization. So basically, looking only at proportions will not give us an answer like whether some subtype increase its abundance or all the rest decrease the abundance. So we want to deal with that somehow. And for that, we developed a method based on simplex spaces. I will not go into math because it's really complicated and I definitely don't have time for that. But the basic idea is that we can assume that if lots of subtypes did not change their abundance and only few did, then probably it's actually these few subtypes which change it. I mean, alternative hypothesis would be that one subtype did not change and all the rest, like, I don't know, are less abundant, and that just has much lower probability. So based on these simplex spaces, we developed a score where for each subtype, we say whether it's overabundant or lower abundant, but it's, of course, all relative to other subtypes. So on the x-axis, you can see this score, and then you can prioritize subtypes which like have higher probability to be actually changed, and you can see which direction it is. And you can also, based on, because you have box plots, you can estimate like confidence intervals, and you can see whether it's significant or not, and all not significant types are below the line, and all significant are above. So you can do this kind of analysis, and it's much more precise than just looking at fractions. And that's one way of looking at things. The other way would be to build a tree and to go top down. So we say that let's take all our subtypes and try to split them into two categories, where one is overabundant and the other is underabundant. And then we can try to do this optimization for splitting the tree and see which subtypes are in each branch. And here is dataset for epilepsy, and you can see that on the lower branch we have all exciter neurons, and on the upper branch we have all inhibitor neurons, and I mean almost all. And it's pretty cool because it's exactly what we would expect from epilepsy, because it's caused by imbalance between inhibition and excitation, and we would actually expect some disbalance between these types of neurons. And then we can go into each of the branches and split them again and again, so we kind of identify groups of neurons which are likely to change their abundance like together, like in the same direction. And yeah, we can look at this tree and try to analyze which cell types are similar according to it. That's it about composition analysis. So let me go further for cluster-free way. Unfortunately, here we cannot operate with simplex spaces because it's too computationally hard and just doesn't work well in terms of math. But what we can do is we can take this dataset, it's from a paper on the multiple sclerosis, and we can estimate densities for cells in control. Like each point here is a cell, and you can see different subtypes, and we can estimate density for cells in control and for, say, cells in multiple sclerosis. And you can see that densities are actually different. You can see that in the middle, this microglia subtype actually changed a lot, and also some difference in oligodendrocytes and in some inhibitor, so exciter neurons. So you can look at that by eyes, or you can do some estimation of density per sample. Then you would again get like a distribution of densities per cell for each of the samples. And you can do some statistical testing on that. So if you do Wilcoxon test, then it would highlight us regions which are changed highly. So you can see again that's exactly one region of exciter neurons, microglia. But what's especially interesting, it's a region of astrocytes. So even though we annotated that as a whole single subtype, we see that there is only one part of it which changed its abundance. So we would not be able to capture it by looking at the whole subtype, but we can see it now in a cluster-free way. So that means that probably next we need to go deeper into this subtype and analyze it further. Then, going to expression shifts, first, what we want to do is to understand like what cell types are affected the most. So here we have data from epilepsy, and you can see blue cells are control and red cells are epilepsy. And to give you an intuition how the method works, what we can first do is to estimate centroids per cell type per condition. So you would have 2 centroids for each of cell types. And then you can estimate distance between these 2 centroids and say that it's kind of a measure. This measure would not exactly work because it's affected by lots of covariates, particularly by spread of a cell type. So you see that if one subtype is super spread, then the distance is of course higher between the centroids, and if they're very narrow, the distance is low. And the same with number of cells, just statistically, the fewer cells we have, the higher distance we would expect. So to deal with it, let's zoom into some specific cell type. And for it, we can take a look at composition of samples, and we can estimate centroid for each of the samples. We can look at their position, and we can estimate all pairwise distances between all controls and all pairwise distances between controls and epilepsy in this case. So we can estimate average distance, average pairwise distance between control samples, and we can take all pairwise distances between epilepsy and control and normalize it on this average control distance. And it can be shown mathematically and it can be shown practically that this normalization actually removes lots of covariates. So it's all like this simple transformation solves a lot of our problems. I will not go into details, so here you just have to trust me, so to say. But at the end, we can get distribution of these pairwise distances per cell type, and it would allow us to get prioritization of differences between the cell types. So we can say which cell types are the most different and which are less different. Next, I want to make a remark on gene expression analysis for case control studies in general, because that's one thing which is really important to keep in mind, and which I never saw in any publication, but we discovered in our analysis. So the point was that we had like 14 samples, and we got revisions from the paper, and we wanted to investigate them further. So we wanted like more data. We got 2 additional control samples, and we added them, repeated exactly the same analysis, and we got really different sets of genes. So of course, what the hell? It's not how you expect it to work, because you expect some stability. Just adding 2 samples out of 14 should not change much. So we started looking at it by excluding samples. So just let's take all our samples and exclude only 1 control sample. So here 1 out of 8, and look at correlation of p-values of genes after the tests. And the correlation is actually really high. So this part looks fine. But if we look at, so we exclude 1 sample, we take we take all genes which pass some p-value threshold, and on the x-axis you see this threshold, and then you estimate Jacquard similarity between 2 sets of genes before the exclusion and after the exclusion. Then you can see that even for excluding only 1 sample, you can get similarity as low, like on average, as 70%. And if you exclude some samples, if you're really unlucky, then the similarity drops to something like 40%. Just excluding one sample out of 14 would remove like 60% of your differentially expressed genes. And that's kind of nonsense. So we still are trying to deal with that. My only advice for now for you is just be careful whenever you do differential expression, especially if you don't have a dozen of sample. Finally, the last part would be expression analysis cluster-free. And the question is, we always do differential expression on cell types, but is there any chance we can do it on single cells and get the genes for each cell? And as you can guess, the answer is yes. And to do that, let's look at the very small region of our dataset. Let's zoom in there. And for each cell, we can define its neighborhood. So we can take k nearest neighbors. You can see them connected to this cell. And we can take in this neighborhood, we can look at control, we can look at the case, and we can take these groups of cells and do differential expression between them. And we can do it for every single cell in the dataset. So it works kind of slow, but still works. And at the end, if you look at the whole dataset, we can estimate z-scores between like for each of cells, and we can just pick genes which affected the highest number of cells, and you can see two of them here. So on the top left, you can see z-score for these genes. Then top right, you can see expression of these genes in the control, and top middle in the control, top right in the epilepsy. And lower is the same, but for the other gene. So you can see that these scores actually show you pretty good the information, and you get the whole matrix of these scores. So you can analyze which genes are changed where without knowing anything about annotation. You can also zoom in for specific groups of cell types, for specific regions, and repeat this analysis, and again do prioritization, and that still works. But what's even more interesting is that if you look, we can take a look at all gene programs. There is a tool called Fabia, which do factor analysis using big clustering. It basically does decomposition of your genes, or in our case, z-scores, and tries to group them and say which of them affect similar cells and in similar way. So you can see program here, and yeah, sorry, just the picture does not match with annotation, but that doesn't matter much here. So yeah, you can see a specific gene program, and you can see genes which correspond to this program. So yeah, this way you can find the most affected regions, and without knowing any about clustering. Again, here is another program, so that works. Another thing we can do in a cluster-free way is doing expression distances. We can do them exactly the same way as below, but instead of using cluster centroids, we do it for every single cell using the same idea of this local neighborhoods. And again, you can see that some subtypes are affected only in specific regions. So L2 CAX2 is affected on the top left, and L56 is F2 LRK1 PCP4 is affected only on the bottom right. So again, you can do this without knowing annotation, and you can look at exactly the most interesting places before doing annotation. So that's kind of it. To summarize, we developed these methods for 4 different types of analysis: cluster-based gene expression analysis, cluster-based compositional analysis, cluster-free gene expression, and cluster-free compositional. And all of them work The work is not published yet, but hopefully will be pre-printed before the new year. And with that, I would like to thank all co-authors and collaborators, and Peter Korchenko for supervising the technical part, and Konstantin Kadushevich Lab, whom I'm a part of, for help and advices. And if you have any questions now, please ask. If you have them later, please email me here. And thank you so much for listening.
Victor, thank you very much for your talk. Colleagues, your questions? Because I'm not bioinformatics, and for me it's difficult to ask any question for you.
Yeah, sure. Can I ask a question?
Oh yes, please, Martin.
Yeah, so it's really nice talk. So I have a question back to the first one you one of your early first ones. So you were able to subtyping the neuron cells, right, more in detail, and then you try to compare the control sample with your disease sample.
All right.
Right. So this, I can imagine this can work nicely for this neuron or like a brain tissue samples. Do you think it's also possible for the oncology sample, like tumor, like earlier I presented, right? So it's still challenging really to subtyping to subtype different cancer cells. So do you have any maybe you have started working on this, or have you looked into this direction already?
Yes, thanks for the question. It's a really good one. Indeed, most of our tests are on neuronal datasets, particularly because our lab is neurodevelopmental, and particularly because there are just lots of them. But indeed, we tested it on one cancer dataset, and it worked. So it does not require you to know extensively cell types, and especially if you do it cluster-free, you don't need any subtypes at all. Mm-hmm. So yeah, we tested on one cancer dataset, and it showed results, so should be working.
Yeah, nice. So do you know how many subtypes of the tumor cells are you identify after for that particular tumor sample?
It wasn't me doing that part of analysis. I would expect something on the scale of 4, 3, I don't know.
And you don't think there will be more different subtypes there? Because what I'm worried is like there are actually many rare subtypes actually is still hidden in the analysis. And I like the way you address this, because it is indeed a problem currently, no matter in which field. Yeah, so that's why I'm curious, like, have you look into this? But yeah, you say it also worked in that particular cancer sample.
Yeah, and sorry, I cannot give details because it wasn't.
Yeah, great. No, that's promising. Thanks.
Thank you.